Hello students, today we will learn the process of the translation. Now see, translation means basically translate. What is the English meaning of the translate? That we are translating it into the different language. Now when we talk about the translation term into the molecular biology, it is related to the translation of the nucleic acid information into the protein part. So whatever information is encoded into the DNA, that is transferred to the messenger RNA. Why it is called as the messenger RNA? Because it understands the code. Now if I don't understand any language, say example I went to some Arabian country, okay, I don't understand the Arabic, so I may have a messenger who will translate the language to me. So after that, that is decoded by the ribosome to produce the protein. So what are the molecules involved into the translation? First we'll talk about the molecules which are related to the translation. First is the mRNA, messenger RNA. Second is the transfer RNA, tRNA, ribosome, enzymes and other factors. So first, say example, this is a small transcript, or you can say the small cistron, a piece of DNA, where the translation is happening. So first what is going to be happen? Transcription will happen. What do you mean by the transcript? Transcript coded information. So that transcript will be formed, and from that transcript one mRNA will be formed. What do you mean by the mRNA? mRNA is a messenger RNA. So the process of transcription will not be discussed into the video. I will make another video for the transcription. If you have any doubt, you can ask me. So after the transcription process is over, after that mRNA will move out. If it is a eukaryote, so in the case of the eukaryote, the mRNA is formed inside the nucleus. Why? Because the DNA is present inside the nucleus. So the mRNA will move outside the nucleus, and inside the cytoplasm the process of translation will happen. So what are the players which helps into the translation? The first and the most important player is the mRNA, which is formed inside the nucleus from your DNA. So DNA is unbound, and inside the messenger RNA will be formed. So it carries the information. Now what type of information? Now DNA have two strand, right? Sense strand, antisense strand. So from the template strand, the same information will be coded from the DNA, or simply you can say we'll take the copy of the DNA inside the nucleus. Now after that it will enter inside the cytoplasm. As soon as it will enter into the cytoplasm, it will associate with the ribosome. Ribosomes, what are the ribosomes? You can simply say these are the translational machineries, or we can say this is something which can understand the information which is present inside. I am telling that messenger RNA has brought a message. So whatever message is been brought by the messenger RNA, that is means this ribosome will decode that particular message. So this will become associated with the ribosome, and after that protein synthesis will happen, putting amino acid into the proper order. But the most important player which helps in doing so, now see this particular ribosome understands the language which is present inside the mRNA, but that language decoding is actually done by the tRNA. It carries the molecule which transfer amino acid to the protein chain, and tRNA is made into the nucleus from a DNA template. Each tRNA is specific to carry one of the 20 amino acid which is used to make the protein. So when we talk about the tRNA, so we have the 20 amino acid which is present inside the tRNA. acid, but we know the 20 amino acid have the 64 codon. So actually we have the 64 tRNA. So tRNA will come to their specific position into the ribosomes, and they will sit attached to their specific amino acid into the pool of the cytoplasm. So I'm not going in detail about the tRNA, because here today's topic we will be just considering about the translation process. So tRNA, first of all, I told you that tRNA, they are going to put the amino acid, they are only going to make the protein. So they will first charged. How they are going to be charged? They are going to charge by picking up an amino acid. So enzyme amino acid tRNA synthesis will help to put the amino acid on that. So example, if it is tyrosine, so tyrosine acyl tRNA synthesis will help and will attach that tyrosine molecule on the tRNA. So for each and every amino acid, one tRNA will be there. A single synthetase may recognize multiple tRNA for the same amino acid. Why? Because same amino acid may have the multiple code. 5' end of mRNA comes together with two ribosome unit. See here the translation is starting. First, so there's going to be three steps: initiation, elongation, termination. Same steps you might remember we have learned somewhere. Think and just message me what where we have studied these three steps. Chalo, let us start with the first and the most important step, that is the initiation step. Initiation means starting of anything. So for starting anything we needs what? We needs energy. So now energy currency in the molecular form is what? It is nothing but the ATP, but here the GTP will be used, and GTP plus protein. So these are called as the initiation factors. So this will bind. Now see, always the initiation start with the AUG. AUG codes for methionine. Okay, so AUG is always the first codon added. tRNA binds to the P side. Now we have learned the three sides: P side, A side, and the E side. There are three sides of the amino acid. Now when we talk about the A side, it is the amino acyl tRNA binding side. P side is the peptidyl tRNA binding side. E is the exit side. Okay, now why the first AUG codon is attaching to the P side, not the A side? I told you that the amino acid tRNA binding side is the A side, but why it is not binding to that side? Because the P side is the peptidyl tRNA binding side, and the first code is going to be the first amino acid, right? First amino acid is what? AUG. So that is present at the P side. So for that particular reason, the first amino acid will bind at the P side, but cholesterol will be binding to the A side and E side. There will be the factors which will be preventing the amino acid tRNA bind to the A side. Means A side को block रखा जाएगा. A side will not be available for the first, that is the AUG, which has an anticodon of UAC to bind. In addition to APE site, there is an mRNA binding group that holds into the messenger site. So see these three actually overall, the large subunit and small subunit, the ribosome looks like this. So these are the three sites. Now next steps is the elongation. Now initiation has happened with the help of the energy. The first amino acid tRNA or amino acyl tRNA has bind to the P site. Now second, what will happen? Now incoming amino acyl tRNA will be binding to what side? To the A side, which is the actual side. It will go. Now see when this will bind. So what will happen? These two amino acid will come together, and when they will come together, after that they will form a peptide bond. Now, why peptide bond? Because now this is the amino acids. The amino acid are going to form the protein. So the two amino acid bind together by the peptide bond. So this peptide bond synthesis will be happen with an enzyme peptidyl transferase. I repeat, peptidyl transferase. Dipeptide is attached to the A side, empty tRNA will shift to the E side, that is called as the translocation. So next lecture I will be continuing with the next step, that is termination. Now for this note we'll stop now because this is the prayer time. Thank you very much.